First, I'm gonna introduce you about rotifer. Rotifer are microscopic aquatic animals commonly found in the freshwater environment such as ponds, lakes, and streams. They play a key role in the ecosystem by feeding on algae and serving as a food source for larger organisms like a small fish and crustaceans. Rotifer are also used as a biodefinicator to assess water quality due to their sensitivity to environmental change. First, I'm gonna introduce you about rotifer. Rotifer. Second, copepod. Copepod are small crustaceans found in nearly all aquatic environments, both freshwater and marine. They are important component of the food web, feeding on phytoplankton and serving as a primary food source for many fish species. Copepod also contribute to nutrient cycling and play a role in the carbon transport in the ocean ecosystem. Lastly is Moina. Moina are tiny freshwater crustaceans closely related to Daphnia, often found in the pond, lake, and rice field. They are commonly used as a live feed in aquaculture due to the high protein content and the fast reproduction rate. Moina thrive in the nutrient-rich water are indicator of organic pollution in aquatic ecosystem.
Zooplankton sampling method. So first we must collect zooplankton sample from ponds and lakes in Yuatemuka using a plankton net. Second is store sample in label collection bottle for transport to the lab. For the material needed is first we need a beaker for sample collection and temporary storage. Second is we need plankton net to filter and concentrate zooplankton. Third is we must have plastic pipette to transfer zooplankton sample. So step 1, preparation. First, select sampling sites such as pond or lakes, ensuring a variety of habitats, open water or near vegetation. Second is rinse the plankton net, beaker and plastic pipette with clean water to avoid contamination. And last, we mark the sampling location and prepare label for collected sample. Step 2, sampling procedure. First, using the plankton net, lower the plankton net into the water vertically, ensuring it reaches the desired depth. Second, pull the net outward slowly to allow water to pass through the fine mesh and concentrate the zooplankton in the net collection cup. Last, if sampling near vegetation, move the net gently to avoid debris while collecting zooplankton. Second is transferring sample. Carefully pour the concentrated zooplankton from the net collection cup into a clean beaker. Next, if needed, add a small amount of pond or lake water to the beaker to preserve the zooplankton. Next, using the plastic pipette, inspect the sample in the beaker and use pipette to pick individual zooplankton, especially for isolating specific species. For example, rotifer, copepod, and moina. Next, transfer pipette zooplankton to separate containers for identification or experimental use. Step 3, handling and storage. Label the container with sample location, date, and time. Store the sample in a cool, shaded area or a controlled environment if immediate processing is not possible. Step 4, observation and identification. Examine collected samples under a light microscope using a glass slide to count and identify zooplankton species. Record the number and type of zooplankton observed for each sampling location. And the last step, data recording. Note environmental conditions such as water temperature, pH, turbidity at the sampling site. Maintain detailed log of sampling activity and observation. Next method is identification and data collection. So first, water quality reading. By using the water quality parameter toolset, take salinity, temperature, and this oxygen parameter. And next is we are going to use design randomized complete block design (RCBD). We use RCBD because this experiment has done in lab. So RCBD must be used for better accurate and better data will get. Record data. Data will be recorded in Excel that have been provided in Google Classroom. There will be 3 different species of zooplankton in Excel that must be recorded depend on their population that we get in lab. The data will be recorded weekly. Next method is data analysis. First, arrange the data. Second, list the zooplankton species and their population in a table. Third is state the hypothesis. Is it null hypothesis or alternative hypothesis? 4. Check assumption. And 5. Make sure the data is roughly normal. Variance between groups are similar. And 6 is we must run one way and a bar. We use software like Excel. So next, look at the p-value. If p-value less than 0.05, there is a significant difference between species. If p-value is higher than 0.05, there is no significant difference. Then the last one is we can report our findings.
Next, we will talk about hypothesis statement for zooplankton community and grow on lake at UiT Muka. They have 2 hypotheses with non-hypothesis and alternative hypothesis. For the non-hypothesis, there is not significant difference between zooplankton community and grow on lake at UiT Muka, and for the alternative hypothesis, there is a significant difference between zooplankton community and grow on lake at UiT Muka. Next, we will talk about the result for zooplankton community and grow on the lakes at UiT Muka. Based on the ANOVA table, we need to reject the null hypothesis and accept the alternative hypothesis. Rejection of null hypothesis and acceptation of alternative hypothesis caused by p-value. P-value for zooplankton community and grow is 1.04 times by 10 power of negative 21, which lower than 0.05, which make we need to accept the alternative hypothesis. 0.05 is the value of the alpha that indicate to choose between null and alternative hypothesis based on p-value for every research. Next, for the conclusion for zooplankton community and grow in lake at UiT Muka, based on summary table, there is significant difference between zooplankton community and grow in lake at UiT Muka. 2 species of zooplankton, rotifer and copepod, was successful to collect the data of population and growth, but the ammonia was the only one species we cannot success to collect the data of population for this species. Moina commonly be found at lakes, ponds, and slow-moving streams where the organic material is decomposing and has poor water quality. While rotifer and copepod commonly be found on any water body which has good water quality. The absence of ammonia in lake prove that water quality on the lake still in good condition.
Next is challenge. First is weather condition. Unpredictable weather, such as heavy rain or strong wind, can disrupt the accuracy of the data. Second, limited equipment. Outdated sampling tool such as microscope lens is cloudy and absent of plankton net limit the ability to collect and analyze zooplankton effectively. And third is time constraint. The process of analyzing zooplankton is taking too much time, especially because it needs to be done manually, which can be a challenge within academic deadline. Future studies should involve continuous sampling during different times of the year. To understand monthly variation in zooplankton population, it is also suggested to analyze water quality parameters such as temperature, pH, and nutrient level to determine their influence on zooplankton diversity. Regular maintenance and or upgrading equipment is also necessary to ensure accurate and efficient data collection. This improvement will enhance data reliability and provide useful insight for environmental monitoring and lake conservation at UiTM Muka. For the implication, this experiment helped us understand the condition of the lake at UiTM Muka by studying the zooplankton found in the lake. Zooplankton can be used as indicators of water quality, which means their presence can show if the water is clean or polluted. The result can be useful for future studies, lake monitoring, and improving water management in the area. To sum it all, the experiment to study the zooplankton in the UiTM Muka Lake faced several challenges, including limited equipment, unpredictable weather, and time constraint. Due to this limitation, two of the objectives could not be achieved, which was to isolate and cultivate identify zooplankton species and to evaluate the growth and population of zooplankton fed with fish meal. Even though not all objectives were achieved, the experiment gave useful information. Zooplankton can show the water quality of the lake, and the result can help in protecting and managing the lake environment better.